This is one most everyone can find interesting, COVID and the microbiome. When we're done, you'll have an excellent understanding about what happens to your microbiome during COVID and how to improve your microbiome and, by extension, your health for the next time you contract COVID or if you still suffer symptoms from a previous infection. Welcome back to another informative presentation with information you'll find nowhere else. I have reviewed all of the available human fecal microbiome data that has been published on COVID. After eliminating a number of original research papers due to various criteria, this presentation will highlight the blended data from 40 papers. The data points have been summed into three logical categories. First, the microbiome of healthy controls as compared to those who contracted COVID, which, by the way, has the most data points. Second, the microbiomes of mild and moderate COVID sufferers as compared to those classified as severe. And third, looking at the microbiomes of those who suffer from what we've come to call long COVID. We'll take a look at what's going on inside the GI tract, explain facets of the microbiome, and look at how the COVID-induced or COVID-exacerbated dysbiosis can lead to secondary conditions such as autoimmune disease. At the end, there will be a protocol specific to the COVID-affected microbiome. Before we get into the effects of COVID on the microbiome, I'd like to review a paper which does an excellent job of highlighting that there are a number of other non-COVID respiratory tract infections which are known to negatively impact the gut microbiome. And although their effects are known, how they come about is unclear. Here we can see how the flu, tuberculosis, pneumonia, and respiratory tract infections negatively impact the gut microbiome, probably in part via the gut-lung axis. We can see that consistently these infections have been shown to significantly increase known opportunistic pathogens within the genera Enterococcus, Escherichia, and Streptococcus. And conversely, they consistently significantly decrease known health-promoting genera such as Eubacterium, Faecalibacterium, Roseburia, and Ruminococcus. Both are the same results seen in COVID, as you will see. These names may be foreign to you now, but will become familiar with you by the time we're done. Now let's take a look at just one of many original research papers analyzing the gut microbiome differences between those infected by COVID as compared to healthy controls. You can see in green the taxa that were significantly higher in COVID patients versus in red the taxas that were significantly higher in the non-infected healthy controls. If you look carefully, you can see that three opportunistic pathogens highlighted earlier with other respiratory diseases, Enterococcus, Streptococcus, and Escherichia, are all significantly higher in the COVID-infected subjects. And likewise, Roseburia, Faecalibacterium, and Ruminococcus are significantly higher in the healthy controls. There are more good and bad actors in the gut, but these are highlighted to give you an idea of a few which are especially important overall and relevant for COVID. When we take into account all of the included studies in my meta-analysis that had data points for COVID as compared to healthy controls, we get the following chart. On the x-axis, we have the various key taxa, which have the most clear data points within the many studies. In other words, the keystone taxa of the COVID microbiome. Keep in mind that any given microbiome can easily have 1,000 or more species. These shown here are an excellent representation of the bacteria that truly make a difference across all disease states. The y-axis indicates the number of original research papers where a given taxon was found to be significantly different. Green indicates that the taxon was found to be significantly higher in the healthy controls, a good thing. The orange color indicates that the taxon was significantly higher in the COVID patients, a bad thing. Not all papers will show significant differences for all bacteria all the time, and it is common to have statistical outliers. But in general, you can see a very clear difference as to which of these taxa are consistently associated with healthy controls and which with COVID. Now, let's take a look at a COVID severity study. If we just focus on one of our identified opportunistic pathogens, the genus Enterococcus, we see that it is significantly elevated for those admitted to the ICU and those diagnosed with bloodstream infections. There were a number of other microbial findings in this study, but the purpose here is to highlight the danger of a dysbiotic gut. The genus Enterococcus has some 22 identified species, but in reality, there are only 2 which come up frequently in the data and are of concern, They are Enterococcus faecalis and Enterococcus faecium. They are opportunistic pathogens. These species and a number of others from other genera are normal inhabitants of the gut. Many of them are in your gut right now. However, as the name implies, they are opportunistic. When the conditions are right, they're able to thrive because the normal checks and balances within the gut are no longer functioning properly. And here, in the case of COVID, with inflammation and gut permeability, whole and parts of bacteria can make their way into systemic circulation where they can cause more grave problems, hence the term bloodstream infection. How ugly are the data points for Enterococcus and its species across multiple conditions? Let's see. It's pretty ugly. The x-axis contains the conditions I've analyzed over the years. The y-axis is still the number of human fecal microbiome original research studies where a significant difference was found between healthy controls and the cohort with a given condition. Orange is still bad and green is still good. As you can see across the board, Enterococcus is significantly associated with disease. You can see data spikes in both inflammatory bowel disease, or IBD, and in COVID. Therefore, in the COVID-affected microbiome, due to a reduction in health-promoting bacteria, a reduction in antimicrobial peptides via COVID's binding to the ACE2 receptor, and other factors, Enterococcus becomes opportunistic and can cause an inflammatory response. Now, let's contrast what we just saw with a genus full of health-promoting species, none of whom you've probably heard of before. Here we have the data points for the genus Roseburia, which, as you can see across all diseases, is almost entirely green, which is to say significantly higher in healthy controls as compared to cohorts with a given condition. You can see spikes in the data for IBD, inflammatory bowel disease, and COVID here. But Roseburia is also often significantly reduced in colorectal cancer and Parkinson's disease. These bars are both a function of the number of studies available for a given condition, as well as the degree of impact within each condition. Let's look at our second and last COVID severity study. Here we see the significant differences the researchers found. I have some new names of bacteria for you. In Figure A are a few new health-promoting taxa shown, which are significantly higher in mild COVID patients as compared to severe COVID patients. As you can see, Eubacterium hallei and Eubacterium elegans and the genus Bifidobacterium all have significantly higher relative abundances in mild patients as shown in yellow, while in severe COVID subjects they are practically non-existent. It is these good bacteria, along with others, that keep the bad actors in check, like Enterococcus. When the good guys are low or absent, the opportunistic pathogens, who are omnipresent, become opportunistic. Now let's take a look at all the data points when compiled for COVID severity. Again, we see spikes in the data for Roseburia and Enterococcus. Just like with COVID versus healthy controls, here in COVID severity, Roseburia continues to be significantly reduced with increased COVID severity. Likewise, the more severe COVID is, the higher the levels of Enterococcus. Here we also see spikes in the data for other amazing health-promoting taxa, such as Faecalibacterium, Eubacterium, and others. These too are reduced with increased COVID severity. Now let's take a look at long COVID. We'll have a series of long COVID slides ranging from 1 month to 1 year after COVID clearance. Here at 1 month post-COVID clearance, these microbiomes had not corrected themselves for these subjects as compared to healthy colonoscopy-controlled data. If you look at Table 3, you see the column for the non-COVID controls, for the COVID subjects who did not take antibiotics, and for the ones that did. If we look at the bad actor Enterococcus faecalis, we see in the healthy controls its relative abundance is very low. And curiously, in COVID it's rock bottom. But in COVID with antibiotics, it shoots up to 3.19% of the microbiome, which is not good for this one species to occupy that much real estate. As a general rule, as seen here, when a bacterium increases in abundance in response to antibiotics, it's an indication that it's not health promoting. On the other hand, we'll look at the amazing 3 species, Eubacterium rectale, Faecalibacterium prausnitzii, and Ruminococcus bromii, all at the same time. All 3 occupy lots of real estate in the healthy controls. All 3 drop significantly in COVID, as we've seen before, and all 3 fall off a cliff in COVID plus antibiotics. Significant reductions in health-promoting bacteria like these are commonly seen with antibiotic use. Now let's look at 3 months post-COVID. Compared with healthy controls, recovered COVID subjects had a significantly higher relative abundance of opportunistic pathogens and a significantly lower relative abundance of beneficial bacteria. Opportunistic pathogens such as Escherichia and Clostridium boultei were significantly increased, while a long list of health-promoting bacteria including F. prausnitzii, Roseburia, and R. bromii were significantly reduced. In addition, as shown here in the slide, Escherichia, that's where E. coli comes from, was correlated with persistent symptoms 3 months after discharge to include fatigue, chest tightness, and myalgia. While the superhero of the microbiome, F. prausnitzii, was negatively correlated with chest tightness after activity, a good thing. At 6 months post-COVID, 76% of these subjects still had at least one persisting symptom of long COVID. In Figure A to the right, you can see them in descending occurrence. Those without symptoms showed a recovered microbiome similar to that of healthy controls. The dysbiotic microbiomes, the ones with the symptoms, included significantly higher levels of another opportunistic pathogen, Ruminococcus navis, among others. And many of the healthy bacteria you're now becoming familiar with, such as F. prausnitzii, E. rectale, and various species from Roseburia, Coprococcus, Eubacterium, and Ruminococcus, were still significantly reduced after 6 months. And to wrap up the long COVID section, this study is 1 year post-COVID. 45% of recovered patients still reported long COVID-19 symptoms at 1 year after discharge. The most common long-term symptoms were cardiopulmonary symptoms, which included chest tightness after activity, followed by systemic symptoms including fatigue and myalgia, and digestive symptoms including constipation and diarrhea. 23% had anxiety and 29% with depression. As for the microbiomes, you can see some of the usual suspects to the right. Now, let's group all long COVID data points together with my now familiar chart. There were fewer studies which looked at the microbiome of long COVID, so there are fewer data points as compared to the other two categories, hence the smaller values on the y-axis. But still, you get the idea. The good guys are still significantly higher in the healthy controls. So what's going on inside the GI tract to affect so many outcomes, such as mortality, bloodstream infection, chest tightness after activity, or fatigue? How are all these connected? The answer is incredibly complex, but we'll highlight one key aspect here and in the coming slides, that of gut permeability. If you recall, when the good guys are reduced in their abundance, the bad actors who are always present in one form or another will begin to thrive under conditions which favor them. These are the opportunistic pathogens. One of the nefarious things they do is either directly or indirectly to cause gut permeability. What exactly is gut permeability? It's a breach in the integrity of the barrier or cells that line the gut, much like a wound on your skin which protects you from the outside world. And there are various ways to measure it. And in this slide we show the results of a study which looked at 3 plasma markers of intestinal permeability in COVID subjects versus healthy controls. In Figure A, you see something called fatty acid binding protein 2. It is an intracellular protein specific to intestinal epithelial cells, and when they are damaged, it gets released into the plasma. As you can see, this marker of intestinal wall damage is significantly higher in the COVID subjects. Figure B highlights the peptidoglycan. Peptidoglycan is a primary component of the cell wall in most bacteria. If you see a lot of it in the serum, it signifies bacterial intrusion from the gut to the body systemically. As you can see, it's also significantly increased in COVID. And in Figure C, we see lipopolysaccharide. Lipopolysaccharide is also a key component of the bacterial cell wall, but in this case only for gram-negative bacteria, which encompass many of the bad bacteria. Again, this serum marker of systemic bacterial burden is significantly higher in COVID. Both peptidoglycan and lipopolysaccharide are referred to as pathogen-associated molecular patterns. That's a fancy way of saying that the immune system recognizes these chemical signatures, sounds an alarm that signals invader, causing a mobilization of the defenses. Although a necessary part of survival, high levels or long durations of immune system-induced inflammation is not a good thing. Another way to measure gut permeability is via plasma zonulin levels. Although the name zonulin sounds like it's from a science fiction movie, it's the name given to a secreted protein in the gut which causes the junctions which hold the cells together to disassemble. In other words, the more zonulin there is, the more your gut has permeability, inflammation, open wounds, which allow all sorts of immunostimulatory actors to go systemic in the body. In Figure B, you can see how moderate and severe COVID subjects had significantly higher levels of zonulin in their plasma versus healthy controls and mildly infected subjects. And to take it a step further, Figure C shows that hospitalized individuals with higher plasma zonulin levels were more likely to die as compared to hospitalized individuals with lower levels of zonulin. In figures F, G, and H, you can see markers of tryptophan metabolism. In this paper, they found that lower levels of tryptophan and higher levels of kynurenic acid were associated with mortality among hospitalized COVID-19 patients. Now, tryptophan metabolism is not a direct measure of gut permeability like the others we've seen, but it is a measure of gut health, which we'll now look into. And yes, markers of dysbiosis are even tied to mortality in COVID. On this one slide, we have 3 key findings which confirm that even among patients with respiratory failure, those with a better microbiome survived. Finding 1, high levels of proteobacteria in the microbiome were significantly associated with increased mortality. The phylum Proteobacteria houses many opportunistic pathogens. Second, the 2 key secondary bile acids shown here were significantly higher in those who lived as compared to those who died. Bile acids, which are in fact hormones, are a decent marker for overall microbiome health. More on this topic in other presentations. And third, as we were discussing in the previous slide, this marker of tryptophan metabolism was associated with survival. If you think my information is too sciencey, I've been taking it easy on you. Check out this slide. The amino acid tryptophan plays a very complicated and large role in our overall and GI health. You're probably familiar with the neurotransmitter serotonin. It's made from tryptophan. There are a number of drugs which focus on serotonin for anxiety and depression. If you recall from the earlier slide, anxiety and depression were still major problems for 1 year post-COVID. In the gut, serotonin plays many roles in gut motility, neuroendocrine function, and inflammation, to name a few. Under inflammatory conditions, tryptophan is shunted into the kynurenine pathway, and this reduces tryptophan availability for other pathways, especially the serotonin and indolic pathways. If you remember from the zonulin slide, high levels of kynurenic acid were associated with mortality, while in the last slide, an indole metabolite was associated with survival. Tryptophan is also a key component of antimicrobial peptides, which are inhibited by the attachment of COVID to the ACE2 receptors. In our last section, we'll look at the connection between COVID and autoimmune disease. There has been a clear increase in autoimmune diagnoses with COVID. In this study of over 641,000 patients, the researchers found a 44% higher likelihood of acquiring autoimmunity for patients who had suffered from a COVID-19 infection. The most commonly occurring one overall was Hashimoto's thyroiditis. Not nearly as common, but the highest risk factor as compared to healthy controls was Wegener's disease and autoimmune vasculitis. In this systematic review, the main diseases reported were vasculitis and arthritis, impacting about every major body part you can imagine. One of the main mechanisms believed to participate in the inflammatory process is molecular mimicry between the viral epitopes and the synovial membranes. In other words, a sequence of amino acids on the virus matches a sequence in one of your tissues. And once your immune system has been primed to recognize the sequence as enemy, it can't determine whether the sequence is the virus itself or you. The authors state that this type of autoimmune response is viral-related arthritis, does not lead to chronic joint disease, and no case in this review was reported as such. Similarly, in this paper, these researchers have identified 28 human proteins harboring regions homologous to COVID-19 peptides that could possibly be acting as autoantigens in COVID-19 patients displaying autoimmune conditions. Again, this is based on the role of autoreactivity via molecular mimicry in response to COVID viral epitopes. In column 1 is the COVID protein that houses the epitope, the amino acid sequence, that the immune system keys in on. In column 2 is the human protein which contains the matching sequence, where the immune system gets confused and attacks self. And in column 3 are the autoimmune diseases associated with this autoreactivity. This manifestation may be more relevant in active disease as opposed to post-COVID autoimmunity, but specifics are unknown. Another similar but different route of developing autoimmune disease from COVID, or in fact simply from chronic dysbiosis, is via the microbiome. Again, we're talking about epitopes and molecular mimicry. Instead of the immune system reacting to an epitope of the COVID virus, it's reacting to one from a bacterium. In all of the research, the genus Streptococcus probably has the most data in this regard. This genus is full of bad actors, and as you can see on the slide in orange, it is significantly associated with ill health in just about every disease state analyzed. Streptococcus seems to play an oversized role in connective tissue autoimmune diseases, where there appears to be a strong matching in identical epitopes or homologous amino acid sequences in human proteins. In a dysbiotic gut with intestinal permeability, whole or parts of various Streptococcus species can make their way into systemic circulation, where the immune system determines that they are an invader, ramps up the defenses accordingly, only to destroy your own connective tissue via friendly fire in the confusion, because some of your own cells are dressed in the same uniform as the enemy. Let's give some perspective to the health-promoting taxa we've seen in the COVID-affected microbiome. This is a slide you'll be seeing often in my presentations. This is the accumulation of thousands of hours of work determining the key players in the microbiome. Collected here are the great universal or almost universal health-promoting taxa of the gut. These health-promoting bacteria have been found to be consistently, significantly higher in the healthier cohorts and significantly lower in the unhealthier ones across all diseases, including COVID. The darker the green, the stronger the data as a health promoter. Out of 1,000 or more species in your gut, this handful plays an enormous role in your health. There are a few health-promoting genera not listed here, like Bifidobacterium, Allostipes, and Odoribacter, which are in other phyla, but these listed here are the main determinants of health in the gut. These incredible bacteria listed here can occupy a lot of real estate and perform many health-promoting functions beyond the highly beneficial one of butyrate production. The blue arrows mark the many taxa shown to be significantly reduced in COVID. In order to increase their number, you have to feed them the fuel they love. By doing so, you drastically improve your microbiome and by extension, your overall health. If you found this presentation informative and you think addressing the health of your microbiome might be a good idea, then in the Protocols tab of my website, themicrobiomeexpert.com, you can find a science-based protocol for this condition, among many other conditions. You're also invited to view and read my testimonials in its respective tab.